And with that, let's kick off our top secret murder mystery workshop for today. So yeah, just as a general overview, this workshop is going to cover some really important bioinformatics techniques as they're used in the forensics industry, and we'll be using it to solve our very own murder mystery case. So yeah, just to introduce myself, my name is Nandana. I use she, her pronouns, and I'm currently a second year at UCLA studying linguistics and computer science.
And my name is Wendy. I am the other workshop lead for this. I am currently a second year at UCLA as well, but I'm studying computational biology. And here are our wonderful, wonderful volunteers. If you see their name and their workshop volunteer background and you have a question, feel free to message either Nandana, message me, message one of our volunteers, any questions you have, or you can raise your hand at any point with any questions at all, and we would be more than happy to answer them and help you out. Another thing to note is if you see these sort of sparkle symbols in the corner, that is sort of later in the presentation, that is where you know, okay, this is where I can get some points. And for those really nice raffles, and you'll see them later, but you'll get them from getting correct answers on the worksheet and or asking and answering questions that Nandana and I will ask you all. And then I will pass things over to Nandana to set our scene.
All right. So yeah, let's just quickly set the scene. Recently, it's been brought to your attention that there's been a murder. The scientists on the case have been able to ID the victim, but they're having trouble figuring out who exactly the killer is. So the evidence we have so far consists of a DNA sample from the crime scene, some photos from traffic cams of people who were in the area when the crime happened, as well as a list of possible suspects. So you, the head forensic scientist at exploretech.la, decide to join the investigation and put your skills in bioinformatics to the test, which pretty much brings us back to where we are today. So to start off, bioinformatics is basically the intersection between biology and computer science. It's a field that really aims to develop software tools to analyze and interpret biological data, and because of this, it's widely used in situations such as forensic investigations. Throughout this workshop, we'll be walking you through 3 techniques that are commonly used in the field, which are going to be DNA profiling, facial reconstruction, and facial recognition in order to solve our murder mystery. So with that, I'll pass it on to Wendy, who will start us off.
Thank you, Nandana. And our very, very first technique will be DNA profiling. The next section is a little bit bio-heavy, so if you have any questions at any point, please feel free to raise your hand so you can ask a question live, or you can drop it in the chat, or if you're a little bit shy, you can privately DM it to Nandana, me, or any of our wonderful volunteers, and they will be able to forward the question over to me. So, like a recipe makes food, your DNA makes you. Our DNA is the recipe book for life. It builds all of us up. So unless you're an identical twin, your genetic code is unique to you. So my DNA is different from Nandana's DNA. And our genome, so all of our DNA is made up of over 3 billion units of DNA that we call nucleotides, and they're represented by A's, T's, C's, or G's. And if you sort of look at this image on the left where you see these little staircase-like things, those are the nucleotides in our genome. And there are 3 billion of these units. That is a lot. If I wanted to read every single letter in my DNA and then compare it to Nandana's, that would take a lot of time, money, and storage. If you have watched the video of Mr. Beast counting to 1 million, it is a very long video. It takes a lot of time. Imagine if we wanted to count to 3 billion, but twice, and then compare the two to see if there's a DNA match. We would be here for a very, very long time. So we will not be doing that today. Instead, we want to look at small parts of our DNA where we can quickly see, okay, is there a match here? Is there a match here? And compare them. So with this, we have what we call short tandem repeats, or STRs, and they allow us to do this. Short tandem repeats are these repetitive sections of DNA, and the number of repeats typically varies from person to person. For example, if, let's say in my DNA, I have AATT repeated over and over 6 times. But if we look at Nandana's DNA, at that place in her DNA with that repeating section, she only has AATT repeated 3 times. When we look at that point of comparison, we can probably make a pretty good guess that we are 2 different people. And if we look at this image on the right, we can sort of see as well that person 1 right here has AGAT, and all 3 people have AGAT at this very first site of repeating DNA. And at that site, this person has 7. Our second person has 4 repeats, and our third person has 12 repeats. So we can make a pretty educated guess that these are 3 different people because the number of repeats is different. Now, let's say that, okay, what if all 3 of these people all have 7 repeats at this very initial site? Okay, well, we're only looking at one piece of evidence, so we can't say for certain like they are the same people. So we need to look at another site like this one right here on the right. Here we also have a repeat of AGAT, but when we look at it, person 1 has 6 repeats, person 2, 19, person 3, 14 repeats. So we can then know, okay, we looked at 2 different places. They're the same in one, different in the other. They're probably different people. So when we're applying this to a forensics case, we need to make sure we look at about 10 different pieces of repeating DNA chunks. That way we can tell if they are 2 different people, because the chance that 10 different sites are identical is very, very rare. So to sort of assemble this DNA profile together, we can follow these 4 steps. The first step is to obtain a sample of DNA. All of the cells in our body have DNA in them. Typically in crime scenes, the DNA we'll find is some blood. Maybe it's a piece of hair. Maybe it's like 2 skin cells. And that's not a lot of DNA to work with. So after we get that, we need to make sure we copy the DNA so we have a lot more to work with. And when we're copying the DNA, that is when we sort of pick out, okay, I'm going to look at these 10 places in it, and then I'm going to assign a color to each of those 10 places. Then after that, we then find the size of each of the fragments. And the way we do that is we have a laser. I don't know if my virtual background will let me. And then when this other arm right here is a piece of DNA, when that DNA hits the laser first asks, okay, what color are you? And then it's like, are you red? Are you green? Are you blue? And then it knows, okay, I am working with this fragment, which goes to this location. And then after that, the DNA, it just kind of reads it. The DNA passes through, the laser reads it, and it's like, okay, I'm getting kind of bored right now. But the longer it takes for the DNA to pass through, the larger the peak we will see in the next slide, and the longer the fragment, the more number of repeats. And then that's how we start assembling this DNA profile. Once we have a DNA profile up and running, we can then look, is there a match between all of the sites? And if there is, we can make a very educated guess that that person is our killer. And I will pass things off to Nandana to show us sort of how to read a DNA profile.
So yeah, as Wendy just said, our last step is to look at our DNA data to see if there's a match between 2 samples in the number of repeats at each location. So if you focus your attention on the image we have up here on this slide, you can see that suspect 1 and suspect 2 have pretty different reads for these DNA sites. And you know that because when you look at the blue lines on this chart, the shapes of them are quite different. Like, for example, the first few lines for suspect 1 are like the peaks are closer together, whereas for suspect 2, they're farther apart. But not only that, if you focus on the numbers that you see under those blue lines, those also have to match. So for suspect 1, again, looking at the very first set of peaks, we see a 14 and a 15 under them. But for suspect 2, we see a 15 and an 18. So even that like contributes to the differences between the DNA profile between suspect 1 and suspect 2. But on the other hand, if we compare suspect 2 and the evidence sample, we see that if you go through each and everything that's on there, if you look at the blue peaks and the numbers under them, you see that everything is exactly the same, which tells us that it's really, really likely that suspect 2 and the evidence sample, all the DNA from that comes from the same person. So before we move on to the worksheet, let's look at one more example. And just to be clear, like if you look at your worksheet, we have the crime scene DNA that you see on this slide is the same as the crime scene DNA on that worksheet. And the suspect DNA is going to be the same as suspect 1 for this. But if we look at these images, we see that there are some things between the crime scene and the suspect that are the same. Like, for example, if we look at the first blue peaks, they have the same shape and they both have a 4 and a 5 under them, but there are some differences. If you guys want to look at this image, can somebody tell me what one of those differences might be? You can either unmute or put your answer in the chat. Either way works.
In section F.
Yeah, I heard in chat, I see in section C and also in section F. Yeah, in section F, like there's a 7 and a 14, whereas the suspect has a 10 and a 14. And the peaks are also like not at the same distance from each other. And in section C as well, while the peaks look pretty similar, again, the crime scene peaks are a little bit closer than the ones for the suspect, and the numbers are also different. It's a 10-14 and a 10-15. So we can conclude that this crime scene DNA and the suspect 1 DNA probably don't come from the same person. So in just a second, we'll put you into breakout rooms to go through the rest of the DNA samples we put on the worksheet. And basically what we want you to do is discuss with each other and find which suspect matches the crime scene DNA exactly in the way that we've looked at so far. So yeah, when we come back to the main room, remember that if you have the correct prediction, that's 1 raffle point for you. So good luck.
And be prepared to share. So make sure you nominate one person from each breakout room who is sharing. And with that, I will open up breakout rooms and good luck. Welcome back, everyone. I hope you all have had some very fruitful discussions in your breakout rooms and are ready to share out who matches the DNA profile found at the crime scene. Let's just go down each breakout room. Where did my breakout rooms go? And then we can all sort of share out. So the breakout room that had Chloe in it, room number 1, who did you all decide was the murderer? And feel free to drop it in the chat if that is faster.
Suspect 4.
Suspect 4. All righty, cool. Next breakout room, which was led by Nandana.
Our suspect was suspect 4.
All right, we have 2 people thinking, 2 groups thinking suspect 4. All righty. And room number 3, who had Haley as their breakout room leader.
We said it's suspect 4.
All right, a 3rd vote for suspect 4. And then room 4, who had Sophie as their breakout room leader.
Suspect 4.
All right, room 5 with Sasha as the breakout room leader.
Suspect 4.
All right, and room 6, who had me as their breakout room leader.
Suspect 4.
All righty. Yes, a lot of you, actually all of us agreed on suspect 4 for the suspect DNA that matches the crime scene DNA, because for that one, a lot of all the peaks, the little blue peaks, all of them matched at all the sections, and for the numbers, all of them, all the numbers ended up matching up, which is very, very nice. Congratulations to you all. And now our next part in our forensic process is facial reconstruction and learning a little bit more about that. We already talked a little bit about how our genes are the recipe for life. Our DNA is the recipe that makes us us. And to sort of talk about this, we can kind of do the reverse. We know that our genes make us. So now we can, actually it's not really the reverse, but we can also now say, okay, I have these genes. What kind of features might I have? And we can do this by understanding the connections between our genotype, our phenotype, and our traits. So our genotype are our genes. That's all the DNA in your body. Our phenotype are the observable characteristics that we have. Feel free to drop in the chat some observable characteristics of humans. Some examples right now are like eye color. Raymond dropped in hair color. Absolutely. Some things that also aren't observable with our eyes would be like blood pressure. That's also an observable characteristic. So many great answers in the chat. We have hair type, nose size, height, detached or attached earlobe, skin color. Absolutely, you all are amazing. And with that, our genotype, the recipe for life, all of our genes, it determines our phenotype, which is what we look like, our observable characteristics. And some of those traits, some traits and some of those phenotypes are determined by a whole bunch of genes, like height. There are hundreds of genes that play into how tall I will end up. But there's also some traits that are just very, very simple. I do not have a widow's peak, I do not think, but it ends up being, it's like your hair kind of goes like that and this little like beak kind of thing. But whether or not you have a widow's peak is determined by one gene and one gene only. So there's a varying sort of complexity behind this. And with sort of predicting the traits, 23andMe has become very popular in the past few years. For those who don't know, 23andMe, you just kind of like spit into a tube, you ship it off, they do some science to it, and then they come back and they say, okay, you are 10% Irish and like 30%, I'm not very good at geography, 10% Chinese. And they'll do like a breakdown. But they'll also have other things in there too. And they'll be like, you probably have like a 50% chance of developing high cholesterol in the future. And the way they can do that is because 23andMe has done a bunch of studies and a bunch of, like, they've looked into it. They've looked into sort of how our genotype determines our phenotype. And they can kind of say, okay, I have this person's genes. There's like a 50% chance they will get high blood pressure in the future. But it's not an exact science. Some traits are harder to predict and some traits are easier to predict. And in the case of our forensic activity, we will be looking at hair color, eye color, and skin color. When we are predicting traits, feel free to drop in the chat sort of which traits you think might be easier to predict, but which traits will be harder to predict. And there are no right or wrong answers with this because this is a very like new field and people are just like, I don't know either. Eye color is one dropped in by Amy. Yeah, eye color, hair color. A lot of things like that tend to be a little bit easier since we're mostly just looking at one thing. But a thing like your blood pressure, there's a lot going on. It's connected to your digestive system. It's connected to how tall you are. It's connected to how much you weigh. So there's so many things that are sort of combining into sort of your blood pressure that it becomes really hard to predict. Thankfully for us, we will just be looking at eye color, hair color, and skin color today, which are a little bit easier to predict. So when we're predicting eye color, also, if you have any questions at any point, feel free to raise your hand or drop your question in the chat. I am monitoring both of them. Our eye color is determined by at least 15 genes. We are still learning more and more about it every single day. And the way we can kind of look at it is we can look at the genes that produce pigment in our bodies. And typically the types of pigment and how much pigment you have will determine your eye color. When we're talking about pigment, we're often talking about melanin. And if you have a lot of melanin, you'll end up with dark hair like me in a lot of cases. So the possible eye colors for our murderer are brown, gray, blue, green, amber, and hazel. So for our suspect, the DNA we found at the crime scene, we took it, we shipped it off to a lab, and now they're coming back to us and they're saying, our suspect has genes for low melanin production. So with that, we can probably eliminate brown, since brown is pretty dark and there's probably a lot of melanin production going on there. The second one is that our suspect has light colored eyes. So that one, okay, we pretty much know it's probably not brown eyes. And our third one is that our suspect has genes for a weak amber gradient. Sort of taking these three pieces of information and kind of like thinking about it, drop in the chat what you think the eye color of our suspect will be. Alrighty, yes, I'm seeing a lot of them filter in. We have a blue, amber or hazel, amber, hazel, 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 amber or hazel, hazel, hazel or amber, maybe blue, green or hazel. We have a lot of things for hazel as well as green. And this is sort of highlighting how exactly, this is not an exact science. So it's kind of hard to fully say 100% like what eye color they have. But the part that we really want to take away from eye color is that we need to use multiple pieces of evidence, as well as sort of like multiple pieces of evidence in determining these things. So for example, this last piece right here where they have like a weak amber gradient, that's kind of signaling to us that it's pretty likely that our killer has amber colored eyes. So, but all of those guesses that you all put in the chat of hazel, of blue, of green, of gray in the chat, absolutely, those are amazing guesses. And for the purpose of this worksheet today, let me switch to the worksheet. Under eye color, put down amber in there. Alrighty, and with that, in your breakout rooms, you all will be predicting the skin color as well as the hair color in your breakout rooms under worksheet part 2. And with that, breakout rooms are open again.
All right, so if our groups would like to share out, we'll go through each of them again like we did last time. So does group 1, let's see. Yeah, does group 1, who I believe was led by Chloe, want to share out their answer? Nothing.
And if not, I'm feeling a little shy. Chloe, what answer did your breakout room have?
Yeah, so we got amber eyes, red hair, and light skin.
All right. So group 2, my breakout room, I know we kind of ran out of time there, but if one of you wants to speak out and share our conclusions, that would be amazing.
Yeah, so our group decided that we predict the suspect would have red hair color and a light skin color.
All right, awesome. Now on to group 3, who is led by Haley.
We said they probably have like a strawberry blonde hair, and I don't think we got to their skin tone.
Okay, yeah, that's completely all right. Let's move on to group 4 then, who is led by Sophie.
We said red hair and lighter skin.
Awesome. Group 5, who is led by Sasha. I see red hair, lighter skin in the chat. Awesome. And yeah, finally, group 6, Wendy's room.
We decided red hair and light skin too.
Cool. But yeah, from our activity, red hair and lighter skin is like the correct answer. But it's okay if you got something different. As we've been mentioning, this kind of isn't an exact science. We have to like predict based off of the information we have. But yeah, now on your worksheets, if you want to write in red hair and lighter skin for the in the chart under part 2, that would be amazing. And we'll go ahead and move on to our last bioinformatics technique. So yeah, our last technique we'll be looking at today is facial recognition. So how does facial recognition really work? And how are those systems built? This has to do with something that we call the training process, which uses a set of data to make our system more accurate. So generally how this works is we would first let the computer make predictions about the data we give it. And if this prediction is incorrect, we would then correct the computer. Otherwise, if the prediction was correct, we would just move on and repeat this whole process until our desired accuracy is really reached. So in terms of facial recognition, that's until our facial recognition system works the way we want it to. So this kind of works like how you would use flashcards when you're studying something. You would typically look at a flashcard, try to think of an answer, and if you're wrong, you'd correct yourself and move on. And if you're right, you'd just move on until you kind of know all the information. So let's see how this training process works with a quick activity. Let's say we're training a model that's meant to distinguish items that are hot dogs from items that are not hot dogs. In just a second, we'll show you a series of images that our computer has classified as a hot dog, and you're going to give me a thumbs up, either physically or on Zoom through the reactions, anything like that. You're going to give me a thumbs up if the image shown is actually a hot dog, and a thumbs down if the image shown is not a hot dog. So we can see like how well our system is doing right now. For the sake of, what was that?
Sorry, I was just going to say, there is no thumbs down reaction on some versions of Zoom. So if you don't have a thumbs down, you can do a cry laughing emoji.
Yes, that also works. But yeah, for the sake of this activity, we'll define a hot dog as just the kinds you can eat. So the two things we want to look for is if the object looks like a hot dog and if it's actually edible. Because even if it looks like a hot dog, but it's not like an edible hot dog, like the kinds you eat, we're going to say it's not a hot dog. So let's jump right into the example. So do we think this is a hot dog? Remember, we're only classifying the food hot dog as a hot dog. I'm seeing a few cry laughing emojis. Yeah, I'm seeing a lot of cry laughing emojis. Would anybody like to share why they think this is not a hot dog? either in chat or like on mute or anything like that?
It's not edible.
Yeah, they're shoes, so they're not edible. So in terms of our computer, we'll say that they were wrong on this one. Let's move on to the next one. Is this a hot dog? I'm seeing more cry laughing emojis. Yeah, this one is still not edible. People in chat, you are correct. It's also not a standard hot dog. Like, well, it looks like a hot dog. It's just a dog. It's just a regular dog, not a hot dog. So this is not a hot dog. Let's move on. Is this a hot dog? Yeah, I'm seeing some thumbs. Amazing. Yeah, not much to say for this one. It's a hot dog. So our computer was right for this one. So that's good because now we'll know that our system was at least a little bit correct. Is this a hot dog? Yep, I'm seeing some thumbs down, some cry laughing emojis. Yeah, well, it does look like a hot dog. It's not edible. It's just on Snapchat, so you can't eat it, sadly. But yeah, let's move on. Is this a hot dog? Yep, more thumbs down, more cry laughing emojis. Yep, this is, while it is edible, it's not a standard hot dog. It's a sandwich. So our computer missed on this one, but that's okay because all of this information will help make our system better. The real question is if hot dogs are sandwiches. Yes, Wendy. But yeah, so let's bring all of this stuff back to how facial recognition in particular works. So the systems that are used in forensics are kind of trained in a similar way, except instead of hot dog data, they're fed with sample data that would be like pictures of people. And then our training process for facial recognition systems would be kind of like testing their ability to detect the faces in the sample data they're given, as well as their ability to match two faces as belonging to the same person. So when the algorithms are actually used, it works in this way through detection, capturing, and matching. So first, our computer detects a face in a photo, and the analog, or like the visual, just like qualitative information about them, is converted into digital data that a computer can actually analyze. Then this data is compared to a list of known faces until a match is generated. And what this really means is that when a match is generated, the computer thinks that the matched faces belong to the same person. So let's focus on part 3 of our worksheet. In just a second, we'll send you off into breakout rooms again. But on your worksheet, if you focus your attention there, we have photos from traffic cams of people who very well could be suspects. And just as a refresher, the description we found for our killer is that they have amber eyes, red hair, and light skin. So what we're going to do in breakout rooms is we're going to kind of go through those pictures and count up how many people we think match that description.
Alrighty, and with that, breakout rooms are open again. Go forth.
All right. Yeah, as we've been doing for this whole workshop, let's quickly go through each group again. And yeah, just when I call on your group, you can have your representative tell us how many people you found. So let's switch it up this time. Let's start with group 6, Wendy's room.
We found 2.
2. Awesome. What about group 5, Sasha's room?
3.
3. What about group 4, Sophie's room?
3.
3. Awesome. What about room 3, Haley's room?
2.
2. What about room 2, my room?
We got 3.
3, awesome. And group 1, Chloe's room.
We got suspect 4.
Nice, okay. So yeah, for this one, as you can see, like in the pictures, there's not really like an exact answer we wanted here, because the kind of point is that like you can't really tell from our pictures, like the suspects or the pictures of our suspects are kind of blurry, like especially for someone like the man on the left-hand side of the very first picture. We can't really tell if he has like red hair or not, just because the image is kind of low quality. But yes, in like the very first man on the leftmost side over there, it's honestly kind of hard to tell. But this kind of exposes a really prevalent problem actually within facial recognition as a whole.
Let me. Okay.
Cool. So yeah, and this is where we kind of have to start talking about bias. So in general, there's many problems that that arise when we rely upon facial recognition as our sole method of identifying criminals. And you kind of saw some of them already. Like if we were just using the data from the pictures and not like any of our DNA evidence or anything like that, it would be really hard to tell like who the killer is, because first of all, there's multiple people that match our description, and also the pictures aren't that great, and that's kind of like how things go in the real world as well. Typically photos like this aren't very high quality, so it's kind of like iffy when we use that as our only method of identifying criminals. And on top of this, the problem of bias is something that is really prevalent, because oftentimes, as I said, when you're making these sorts of systems, you have to train them on pictures of actual people. But what usually happens is that some groups of these people are often underrepresented. And what that really means in the long run is that like if one group is underrepresented in the training data, the facial recognition system will be worse at identifying them. So when we compare women to men, women are often underrepresented in training data. So facial recognition usually works better on men. And the same thing goes for people of color to white people. And this has like honestly really like severe implications at times, because there have been many incidents in which like when police used facial recognition to like kind of find a suspect or like to arrest someone, there have been instances in which like Black people have been wrongfully arrested because the facial recognition system was just not good at identifying Black faces. So when we're relying on this sort of like one-to-many kind of verification, it kind of can result in false positives a lot of the time and lead to instances wherein people are like wrongly arrested. And that really begs the question of how much we should be relying upon facial recognition and systems such as these to match suspects. So let's move on. And yeah, here is a quick look at like all of our suspects. We'll send you off into breakout rooms again. And in these breakout rooms, what we want you to do is to take a look at all of them and discuss all of the evidence we've gotten from the first 3 parts of our workshop so far. And based on all that evidence, see if you can come to a conclusion on who the killer is. So yeah, as always, be sure to nominate a person who will share out when we come back. And yeah, we will have about 2 minutes for this room, I believe.
Yep. And with that, breakout rooms are open. So group 1, led by Chloe, who did you all identify as the killer?
Suspect 4.
Alrighty, thank you for sharing. Breakout room 2, led by Nandana.
Suspect 4.
Great. Room 3, led by Haley.
Suspect 4.
Awesome. Room 4, led by Sophie.
Suspect 4.
Awesome. And then room 5, led by Sasha.
Suspect 4.
Alrighty. And room 6, that was led by me.
Suspect 4.
Awesome. And then quickly in the chat, feel free or unmute yourself and share. All of you, what were some sort of pieces of evidence that led you to point towards suspect 4?
His DNA was at the evidence, and then he's the only one who had like all amber, red, and light skin.
Absolutely. Thank you for sharing, Raymond. And then Marin said the DNA match. Sophia said DNA match. Natalia said facial features. Angie, eye hair color match. DNA predictions. Absolutely. You all correctly guessed the murder, which is absolutely amazing. And this is sort of a bit of an overview slide on why suspect 4. So like you all mentioned, the DNA matched, the heights of all the peaks matched, suspect 4 to the crime scene DNA, and the number of repeats matched as well. When we were talking about facial reconstruction, suspect 4 we determined has amber eyes, red hair, light skin. Sorry, I switched it up. Our murderer from the crime scene had those features. And when we look at that suspect bank, suspect 4 has all those features as well. And then lastly, with our facial recognition activity, though we weren't able to say like, I know suspect 4 right there, but we were able to identify at least, I believe the lowest number was 2. We were able to at least identify 2 people in those photos that had amber eyes, red hair, and light skin that were present near the crime scene. So sort of using all of this and like squishing it all together, we can make a pretty educated, pretty informed guess that suspect 4 is our killer. And with that, congratulations to you all. Thank you for being such amazing students and for going through this workshop with us and learning a little bit more about the wondrous world of bioinformatics. And congratulations on catching the killer and making sure that Alex gets the justice that Alex deserves. And with that, you all are not free to go yet. We are taking a group photo so that I have a group photo. So if you all, also for like content capture purposes, but if you all are able to and comfortable, please turn on your camera. If you're not, that is perfectly okay. You can do one of those happy face reacts in the Zoom reactions panel. All righty. Oh, there's 2 screens of people, but I will take multiple photos. All right, 3, 2, 1. Okay, now let me grab the second panel of people. All right, 3, 2, 1. Whoa. Okay, I have my screenshots. Cool. Thank you so much, everyone, for being here, for participating. I could not have asked for a better group of students. Nandana, are there any last words you want to say before we let them roam free?
No, thank you all so much for being amazing students and coming out to this workshop. I hope you learned something new. I hope you had fun during this workshop too. Thank you. Yeah, thank you so much for coming out.
And with that, you are free to leave. Have a good rest of your day, and I hope to see you at some of the workshops tomorrow.